All right, so we're here with Dr. Judy Anderson from the University of Manitoba, and she is a researcher in satellite cell research, focusing on muscular regeneration, satellite and stem cell biology, and human muscle disease pathophysiology. Dr. Anderson, thank you for being here.
You're welcome.
So I'm pretty excited to go into this whole discussion on satellite cells. I want to tell you, and I've told you already, but I'm going to tell you again, I've had this, deep interest in this topic since I was in middle school, just because I was fortunate enough that I had a dad who was interested in strength training and got me into different aspects behind research. And at the time in the late 90s, early 2000s, there was research on satellite cells, but I was 15, 16 years old and I was an idiot and I didn't have a full understanding of what exactly they were and how they can impact muscular regeneration. And so I guess I want to start with you is what exactly are satellite cells? Where do they live and how do they operate?
Okay, so satellite cells are something I stumbled on in my research, more or less out of an anatomical perspective, because my training was in human anatomy. So I love muscles and bones and how they fit together and work. And so my research kind of spans interest areas of exercise and anatomy and pathology, what makes things go wrong. And then because of that, an interest in how things work, like at the core. Mm-hmm. So satellite cells are named because they're kind of like a little satellite. They kind of orbit and move up and down a muscle fiber. So they're very tiny, and a muscle fiber can be anywhere from like microscopic length, a millimeter or less, to 6 or 8 inches, depending on the muscle. So in your biceps muscle, a muscle fiber isn't the whole length of your muscle, but it might be 3 inches, 4 inches, depending on the person. And the interesting thing is that in mammals, and people are a mammal, you can't make additional muscle fibers unless you're repairing muscle. So no amount of exercising will make the number of muscle fibers you have increase, at least not very much, unless you do enough injury to them that they will form new fibers in parallel with fibers that are going to be repaired.
And that's post-mitotic, right?
That's post-mitotic. So satellite cells in muscle are the only muscle cell that can divide, because all of muscle fibers are made up of fused muscle cells that can no longer divide. So when they commit to fusing and making muscle, they stop being able to divide. Okay, so we got a few satellite cells on a big muscle fiber, and there's many muscle fibers in a muscle like the biceps or quadriceps has thousands of muscle fibers. Right. Now you need to make that muscle fiber grow. So growth happens 2 ways. The nuclei inside the fiber can expand how much cytoplasm or protein is around them, which is the mechanism of making muscle work, so that the muscle fiber will grow in diameter, or new satellite cells on the outside of that fiber will divide, and some of them will fuse into the fiber, and those new nuclei will grow, like the cytoplasm around them will grow. So the first way is called hypertrophy. You have an expansion of cytoplasm around the nuclei in the fiber. And the second mechanism relates to the hyperplasia, or division of satellite cell progeny, because satellite cells are quiet. They don't really divide. So let me show you what sort of a model of a muscle fiber. would look like. So we've got a muscle fiber. Generally, they're just depicted as a cylinder that goes from, say, one tendon or bone attachment to another. So the muscle fiber is going to stretch and contract in that lengthwise dimension.
Mm-hmm.
On the outside of the muscle fiber are satellite cells. This is a really wimpy satellite cell, quick model. They're very thin, little tiny cells. This is way too big for this muscle fiber. And they are outside the muscle fiber. So we knew for many years before I started in muscle research, we knew that this was the cell, the nucleus in this thing was what divided. The nuclei inside this muscle fiber, they can't divide. Okay, they're post-mitotic. This can still be mitotic. And many people had studied muscle growth, muscle development, how satellite cells make muscle in the embryo, or how muscle cells make muscle fibers in the embryo. And then in the early to mid 90s, people learned the genetic regulatory factors that would control cells becoming muscle. And they started to look at the expression or the production of those proteins that regulate muscle quite soon after an injury. And always in that time, the goal of treating muscular dystrophy drove a lot of this interest.
Okay.
Treating disease often drives interest in research. So what people learned was that, within 12 hours, or certainly within a day of an injured muscle fiber, those regulatory genes were turned on, and those cells were going to start making more satellite daughter cells or muscle cells that would divide and make new muscle, regenerate muscle fibers.
And that's essentially how a human's body is going to be healing a site of trauma.
Right. So my interest was, well, what actually starts that whole process off? What starts the process that makes those satellite cells go, oh, there's something wrong here in the muscle fiber that I'm sitting on?
Right.
So, I always was interested in what started that. I was interested in all the other steps along the way that would make new muscle, for sure.
Right.
But I was always interested in what started it. And then I started to think that little tiny thin cell might be adapted to feel some kind of signal from the muscle fiber right under it.
Right.
And I used to liken this to a fried egg on a sewer pipe.
Okay.
Because that kind of gave the scale. A little thin flat thing with a bulge, nucleus,
right,
sitting on a great big huge cylinder. So what would give you a signal could be, you could think of something like a vibration or a physical change in the muscle fiber.
Even in your one paper, you described it as a perturbation that initiates the whole system of communication, essentially.
Right. So I was looking for that perturbation. And then I started to get interested in this molecule called nitric oxide synthase, which is a molecule that synthesizes a gas called nitric oxide. And it turns out that NOS is mechanically sensitive. So, what tissue do you know that is mechanically based? Like muscle and bone and skin are like totally mechanically
no matter
geared in.
All movement based.
All movement, stretching, twisting, everything. So then I started to think about the possibility that nitric oxide synthase was known to be in the membrane of muscle cells.
Right.
and it was known to be mechanically active. It turns out nitric oxide was the molecule of the year one year. You know, people were Nobel Prize things were happening. I knew nothing about that when I started to think, well, maybe nitric oxide has something to do with this signal coming from a muscle fiber and getting to a satellite cell. And I thought, what better way to receive a signal than to be holding on to a sewer pipe that might give you a little flash of something.
Okay.
Right?
Yeah.
If you're going to be set up for that. It turns out, well, of course, it's never that simple, but it turns out that there is, when muscle is stretched or does work, there is a release of nitric oxide gas really fast, within milliseconds, from that muscle fiber. And that signal initiates the release of this other molecule called hepatocyte growth factor, HGF we call it, which is stored around in the matrix around muscle fibers. And that just happens to be the protein that binds to a molecule in satellite cell membranes called the c-Met receptor. And that receptor turns out to be known as what's motogenic. In other words, it's like a motor. It makes cells move. So cell movement to be, those satellite cells are normally just there, quiet, on the membrane. But when they are activated by mechanical stretching or nitric oxide, they start to move. So their movement signals the beginning of them entering into some process that makes them start to cycle and divide and then contribute new muscle cells, either to the fiber close by that needs repair or to making a muscle fiber.
So that's where my next question would be then, is it's satellite cell stimulated by NO, nitric oxide synthase, and
right.
HGF is part of the whole scenario. How does it know where? How does the satellite cell know where to go? Does it also, in that signaling, is it recognizing where that trauma is, or is it just that where that perturbation is, that NO it releases, and then it basically tells the satellite cell it needs to arrive there and heal?
Well, I think it's actually both.
Okay.
And in fact, another arm of the current research I'm doing with a student is trying to think about what makes things go in a certain direction, what makes muscle cells go the way they go, more or less. Just a sec. So if, remember I said the muscle fiber is surrounded by a matrix of proteins.
Right.
Some of those are things like collagen and fibronectin and tenase, various proteins. So laminin. So that the combination of molecules in the direct area will influence how quickly something might move or which direction it goes, because just around this one cell there might be, especially if this muscle fiber is injured, a change in the type of protein that coats this fiber because of the things that are released from a damaged muscle fiber. So they may change the matrix around the muscle fiber and pull the satellite cell in a certain direction once it's activated, or repel it and push it away so that it makes new muscles at some other location. So it turns out also that because HGF links to that receptor on satellite cells that is motogenic, that HGF is called a chemokine, and it can actually speed up the migration of satellite cells and help direct it to a certain place. So a high level of HGF can be a positive stimulus to the muscle cell as it's moving.
So where exactly, I'm trying to think here as far as if we would think about, because in the one article you sent me, you talked about IGF-1 and mechano growth factor, and then so I'm trying to, in my head, I'm trying to piece together, where does IGF-1 and mechano growth factor come into play, and where is PAX7 and PAX3 just a sign that it's that satellite cell has been activated?
No, PAX7 and PAX3 are very interesting genes because they kind of specify the lineage that you're going to be muscle.
Okay.
So the satellite cell has PAX3, 7 all the time. Muscle fiber nuclei don't. They did when they were single cells, but once they fuse and make a muscle fiber, they no longer do.
Okay.
And so IGF and mechanogrowth factor are present, and they're made by muscle. So they're made by muscle fibers. And it turns out if you culture single muscle cells in a dish, then it's also made there. And it also stimulates their growth. So doesn't want anybody to think that HGF and nitric oxide are the whole story. They're certainly not. It's just that they're at the very front of the process.
Okay.
And after that, there are many, many signaling pathways that take that activated cell and determine how quickly it'll divide, how many times it'll divide, where it'll go, when it fuses, how quickly it'll grow as a muscle fiber, all of that sort of stuff.
And that what you just described is essentially why you would call it the currency of muscle regeneration is a satellite cell.
Yeah, because it's kind of like the building block.
Okay.
Right. You need them to build new muscle for sure. And you certainly need them to repair muscle fibers. And although it's not the same kind of cell in development, you need building blocks, and they don't have a satellite position, but they're all muscle precursor cells.
Okay. So in regards to satellite cells, one of the things, and I don't know if this is accurate, I've always, in my mind, I've thought there was a finite number of satellite cells to a point in the reservoir where they're sitting. Is that accurate or is that not accurate?
Yeah, there's usually a number, like each muscle fiber will have a population of satellite cells on it. And depending on what kind of muscle fiber you are and what animal you're in and how much stretch that muscle is usually undergoing, you'll have a sort of representative population. So just as an example for context, maybe if I look at a mouse fiber that's maybe say up to 1 millimeter long, it might have 6 to 11 satellite cells.
Okay.
If you're a muscle fiber from a muscle that's mostly like a postural muscle working pretty much all the time, you're the word slow fiber. Right. You're doing work, you're a red muscle.
Right.
There are more satellite cells per fiber in a slow fiber than there are for a fast fiber. And fast fibers are usually larger in diameter than slow fibers. So if you think about trying to find them, if you want to isolate satellite cells from a muscle and you don't care what they come from, you pick a muscle that's working hard most of the time, like a slow fiber muscle.
Okay. So what, is that reservoir, like that finite number of cells, is that consistent upon like, hey, my erector is going to have the same relative number of satellite cells as your erector would, or does my body's, before, someone once did explain to me that up until you hit puberty, your body's essentially preparing for the rest of your life based off of what its perception of what your perception of physical struggle will be. So your satellite cells, if you are, and I like to use this example of farm strength. If you grow up on a farm, you might have more activation or a larger pool of satellite cells to pull from than if you grow up, in a much more sedentary existence up until puberty. Would that be accurate or no?
I think it would to some extent, but I have to say that if you're growing muscle while you're growing up, and as an adult, young adult, you're into building muscle, that you're probably also stimulating those satellite cells to divide and fuse with the muscle fibers that they're on or nearby. And you end up with a lot of nuclei in the muscle fibers because you've got big muscle fibers.
Right.
If you're healthy, doing it in a healthy way. Then I know there's work that shows that if you then become, let's say when you're in your 40s or 50s, a bit more of a couch potato, your muscles will atrophy some. But it takes quite a lot of atrophy before you start losing nuclei.
Okay.
And then if you go back to exercising,
sort of wakes
the
up.
Nuclei in your muscle fibers can respond pretty quickly to exercise by growth of the cytoplasm around each nucleus. This doesn't mean you're going to be a bodybuilder again.
Right.
And the older you are when you get back into exercising, the harder it is to keep growing muscle, but you will feel more fit.
So would that be almost like, I mean, I'm thinking about in my case where I coach, shot putters. I have a girl last year, she was ranked 13th in the world, and she tore her Achilles, about 60 days ago, actually today. So in that case, the individual who did surgery on her just kept saying like, hey, you're going to recover well because you're an elite athlete, but two, you're also going to get your movement back in the circle. You'll have muscle memory. Is that like the scientific?
Yeah, that's what this author has spoken about.
Okay.
Who talked about the myonuclei having, essentially they're banking the potential to grow more muscle cytoplasm, the stuff that does the work, by staying in the muscle fiber, even though the muscle fiber itself might shrink in mass and diameter some. Now, up to a point, right? Because if you never go back to it, or suddenly you lose the ability to do any voluntary movement, like you're in a car accident and you're paralyzed, those myonuclei won't stay there forever. They'll gradually be lost, and you'll have fewer fibers.
Okay.
Fewer nuclei in each fiber.
Okay. I guess so another question, going back to the beginning when we talked about some of the stuff regarding hypertrophy and hyperplasia, I was always sort of under the impression that there's myofibrillar hypertrophy, which is like regenerating the myofiber specifically, and then there would be sarcoplasmic hypertrophy, which would be that cell expansion without increasing the number of myofibers. And is that an accurate portrayal of hypertrophy? And then if so, can satellite cells impact sarcoplasmic hypertrophy the way that they, or in a different way than they do myofibrillar hypertrophy?
Okay, so the way I speak about it is within a muscle fiber, there are many myofibrils.
Yeah. Yeah.
Right?
Yeah.
And that is really the cytoplasm of the muscle fiber, which is doing all the contraction. Myosin and actin proteins in sarcomeres from one end of the fiber to the other. And each nucleus in that fiber will make proteins that contribute to the myofibrils and all of the other mechanisms inside that muscle fiber. So myofibrillar hypertrophy means each myofibril inside there, like a whole bunch of fingers, each one is a myofibril,
Yep.
and they're in a fiber. Each one of those will grow based on the signals that each nucleus around them gives them to make new protein, make more protein. And then if you want the whole fiber to grow, then essentially pretty much all of those myofibrils will be expanding. So myofibrillar hypertrophy or growth is more or less the same thing as sarcoplasmic hypertrophy. You're going to get more volume of each fibril, so your fiber gets bigger.
Okay.
The satellite cells do something else. Out here you have a satellite cell. Let's say it divides. You get two, and one of them fuses in. Now you've got the possibility of either starting another fibril, maybe, because they don't only make one, but it will contribute more cytoplasm inside that muscle fiber, so it can grow a bit more in diameter. So the satellite cell contributions through proliferation will make that fiber grow because you're also increasing the number of nuclei.
Okay.
If you don't have satellite cells contributing, then the growth is the same expansion of the fibrils or the protein, the sarcoplasm, but you don't have an increase in the number of nuclei inside the fiber. So you can't grow more and more. You can only grow up to a point because it's like how many square feet of house can one person clean,
Right.
manage, support, pay taxes on?
Yeah.
If you want more square footage of your house, you either got to have another person join in to help pay,
Yeah.
right? Or what? I don't know. You got to have more people pay it, right? So that's why it's like the currency. You can grow quite a bit without new satellite cells contributing to your muscle fiber. Quite a bit. Like I don't even know what that amount would be. Double, triple, quadruple. You can grow a fiber a lot because there's a huge amount of potential for growth, but up to a point. And if you want it to keep growing, you need more nuclei to make more protein inside that muscle fiber.
And that nuclei is donated by the satellite cell.
Yeah. It's a satellite cell when it's right there on the periphery. Once it's sort of activated and dividing, then it's called a myoblast.
Okay.
And then it keeps going. But those myoblasts are called the daughter cells of satellite cells.
So going back to the activation of the satellite cell, is it because you're talking about the lengthening of the muscle fiber? Is it in theory? Is it the longer and does this go back to the eccentric movements of resistance training? The longer an eccentric exercise, or the longer the eccentric contraction is, the more that muscle fiber will get stretched, and in theory or practicality, the more satellite cells will get recruited because of that lengthening of the fiber.
So I really only know about that in relation to aging, but the short answer is yes.
Okay.
So when you're young and you give a certain amount of stretch, let's say you lengthen the muscle by 10%, you'll activate X proportion of satellite cells.
Okay.
If you are older and you stretch the same amount, 10%, you will activate fewer, or you won't activate any at some point.
Mm-hmm.
So older folks need to exercise harder, more resistance to get the same
response.
Activation. And then whether you have the same growth is a slightly different issue, right? Because it's not just the nuclei, it's the ability for those nuclei to make a lot more new protein.
Right.
Make all those contractile myofibrils and everything else inside the cell.
So I can't remember if we were actually live and recording when we were talking about this, but when you brought up that you've done research or you've seen research with I think it was NOS and completely immobilized limbs, and that NOS can actually get a response, a muscular growth, without even adding in a form of resistance or stimulation to the muscle area.
So we were able to show reasonably recently that in an animal where you immobilize the hind limbs you suspend a mouse from its tail, it still can move on its front legs, but its back legs don't hit the ground and they can't touch anything. If you administer something that will donate nitric oxide to the body, then you can significantly prevent the atrophy that will happen when those hind limb muscles are not used. So normally muscle atrophies very rapidly, especially muscles like the quadriceps. They will go rapidly shrink if you have sore knees, right? You will protect those knee cartilages by not working that quadriceps, because it pulls on the knee joint, and you will rapidly lose muscle mass in your quadriceps. So we were able to really slow down the atrophy a lot.
just from the NOS being administered or the donator of
the donor of a nitric oxide molecule. Yeah. Okay. Now, that it's not to say that it's only going to muscle. So, we know that when you do that, you stimulate muscle fibers, because we've done isolated muscle fibers and studied activation. But when you give a whole animal that's alive nitric oxide, we also know that it does something to your circulation. It does something to your therefore your cardiac resistance, and all kinds of things are changing. But we know from many experiments now that when you administer this nitric oxide donor molecule, or another one that we had designed, that you can stimulate muscle satellite cell activation and reduce muscle growth, atrophy, and stimulate growth from exercise.
That's pretty cool. That would be huge. In mice. Now, that'd be something that you could do in theory if somebody's immobilized for they break their femur and their legs immobilized for 3 to 4 months. Well, now if we would use, I think it was ISDN.
Yeah, if you could start like doing your rehab before you came out of the cast,
that'd be huge.
Even with isometrics inside the cast, mm-hmm. right? Which some people tell you to do. And then you were able to have some, theoretically, there's reason to think you would be able to stimulate muscle growth or prevent the atrophy that you have inside that cast.
Right. So
I always wanted to do that experiment, but I'm a mouse doctor.
and finding people who'd be willing to do that would probably be pretty tough. Like go sit in a cast for 4 months.
No, I expect it wouldn't be that hard, because it doesn't take long to lose your muscle, right?
Yeah.
In a cast.
That's true. That's true, actually. Yeah.
And you know how if a person who's 8 breaks their femur or their humerus, and they're in a cast, and they take the cast off, and they got this wimpy little arm, right, or leg.
Yeah.
And a week and a half later, many people won't be able to tell the difference between then and before the break.
Right.
Because young people respond so fast.
Yeah.
They get right back at it. And older people take a long time. And I mean, it gets scary when you get older, like I have, and well, we all do, hopefully, because you realize that time isn't something you want to mess with. And
yeah.
you know, you want to be able to get right back at it. So, but you actually have to work at rehab differently when you're older than when you're younger. You have to work at it.
Right. And that also goes back to your research with the satellite cells. Is like, it takes more stretching and more stimulation to get the satellite cells to activate. And that sort of brings me a couple of the papers mentioned myostatin, and I have like a very brief, very generic understanding of myostatin, and one of those is because one of the guys I trained with had a heart condition, and he had a lesser level of regulatory myostatin than I would have, and he's this huge guy, just this guy, his head's like enormous, and he's had this heart condition since he was born, but he's also just extremely muscular. And I wanted to bring that up, and how does that downregulation of myostatin factor into the satellite cell proliferation?
Yeah, it turns out that myostatin has an effect on satellite cell behavior and how much it will divide given the signal, but it definitely affects muscle growth. And so it kind of has a dual influence that way. In fact, many of those growth factors and possibly myokines will affect the satellite cell compartment or group of cells differently or slightly differently than the muscle fibers. And so when you read the literature, you kind of always have to keep in mind that the effect that's measured might be an overall effect on muscle strength or flexibility or diameter, but it doesn't always separate or allow you to, as a reader, separate what's happening to the satellite cells compared to what's happening to the fibers.
Right.
So you could have a stimulus that, let's say, theoretically made muscle satellite cells divide like crazy and migrate a lot, but didn't do anything to regulate their fusion. And maybe when they fused, they didn't fuse in an organized fashion or systematically throughout the muscle. Maybe it only fused in a certain place. And that would give you quite a different outcome than something that stimulated growth of a muscle fiber itself without involving the satellite cells.
Okay. So, I mean, I got a couple questions just off of that, where it's like muscular growth. So do you have to have satellite cells present to get muscular growth?
No.
Okay.
But at some point you need them.
Yeah. There becomes like that point where of diminishing returns, which is like, okay, someone who is a more, I mean, if we're talking about resistance training, if you're in resistance training for a year, maybe up to that point, it's adaptations of coordination with the nervous system, but after a year, you have to basically start to stimulate, whatever that time frame would be. You have to start to stimulate.
Yeah, I doubt that it's a year, but I'm pretty sure that the whole goal of resistance training, at least in older people, is that you build up the stamina to be able to adapt and respond and coordinate with the nervous system and balance more effectively. And so it's not just keying up the growth aspect. Certainly the coordination is important. I should say that another aspect of what satellite cells do is, if you if you've thought about satellite cells at all, which many people don't, you can think of them as this passive thing stuck on the outside of a fiber waiting to be woken up to do something, right?
Right.
But it turns out that if you think about satellite cells as directing cells around them to do certain things, then you unveil another role that they might have. So a collaborator of mine in Japan and I and our labs combined have looked at something that satellite cells make once they're activated, called semaphorin 3A, which is known to repel the ingrowth of nerve fibers, nerve axons. And it's thought that the timing of when they make this semaphorin may regulate the speed at which nerve fibers grow into muscle after an injury to muscle or nerve or both. And another arm of the research that's going on now in the lab is looking at that molecule in relation to satellite cells and cells around nerves. And it actually turns out that there are more satellite cells around the location where the nerve axon comes to the muscle fiber than other places along the fiber.
Okay.
So it shows you that not all satellite cells are equal. There's different types of them.
I think that what you just said too, it also sort of describes when you were saying that the satellite cells are mainly like a director of other cells. It gives you that thought that, for a while, everybody, for the last 100 years, people have thought of muscle as just like this dumb thing in the body. But in reality, it's an organ, and it's responsive, and the myokines are interacting now with, and satellite cells are sort of coordinating everything that's going on. And it's,
well, yeah, not everything, but a
but
fair bit. Yeah, they're making a directing contribution, not just a passive contribution.
Right, and I think that most people have never, they don't think of muscular adaptation in that sense.
No, probably not. But I have to say, I think that's coming more and more.
Right.
And one of the things that I found very interesting is that when you think about satellite cells in people or mice or mammals, you learn the literature in one way, and that if you start thinking about muscle in animals that you might know, maybe you eat them, or maybe you see them running across the field or swimming in the water, it's different, and there are many variations on a theme, so to speak.
Right.
Some of our recent work, we've looked at lamprey muscle. Well, lampreys are ancient animals. And it turns out that although for ages, like over a century, people have known about how muscles in the head of the larva are one shape and muscles in the head of the adult are different, they undergo a different remake, like a metamorphosis between larval and adult phases. There's very little known about what's happening in the body wall muscle. It's kind of assumed that it's not changing much, even though it's known to continue to grow.
Right.
So we looked at muscle there, and I didn't find other descriptions of things called satellite cells, but we found small cells outside muscle fibers that express those muscle regulatory genes I talked about.
Right.
And we've just published a paper on sturgeon muscle. And sturgeon is another thing. They come from an egg and a sperm, and they grow huge and they live 100 years or more. So how do they do that? How do they keep
What's their adaptation to be that successful?
So they make a lot of new muscle fibers.
Right.
So we started to look at what satellite cells do there.
Yeah.
And we found evidence that the cycle of the satellite cell proliferating changes depending on your age, that the population of satellite cells changes with age, with the diameter of fibers, with the length of fibers, with how pointy they are in the ends.
Yeah.
Like we just opened the door a little bit and we found all these neat questions to ask. And like there's a lot of kinds of muscle in the world.
Right.
Tons.
I guess it's like the more you're researching this, the more you're like, wow, we really don't know a lot.
Right. But when you eat meat, you're eating muscle.
Yeah.
So your tongue and your brain can tell different kinds of muscle, texture, taste, blah, blah. There's a whole science of meat that relates to muscle growth.
Right.
Right?
That's crazy.
Yeah.
I have one not related to sturgeon, so I apologize for not diving into the sturgeon research. But when you mentioned that in the first emails when we were going back and forth, I did look at some of that. I didn't read it as in depth, but I thought it was neat. So some of the research too that I've been interested in is and this goes back to the stretching of the muscle and sort of the activation. And I guess this goes along with resistance training. If you have somebody who's doing full range of motion squats, they have a longer eccentric movement. There's a ton of mechanical action going on, so a lot of perturbation for the satellite cells to be activated. And research shows that if somebody does 6 to 8 sets of back squats, they'll have higher levels of testosterone, for the next 48 to 72 hours, and they'll have higher levels of IGF-1. And this is sort of where they think contributes to muscle protein synthesis. And so how does that scenario, play into, and that's in comparison to somebody who might only be doing 2 or 3 sets.
Mm-hmm.
They would have a lower level of IGF-1 and a lower level of testosterone. How does that, with the model of the satellite cell explanation, how does that, where does that testosterone increase play into proliferation or mobilization of the satellite cell?
Yeah, so I think the testosterone would be acting on the muscle fiber.
Okay.
And the IGF-1 could act on increasing the number of, I don't think it would increase the rate of satellite cell division, or it doesn't change activation itself, but it promotes proliferation. It might help keep the satellite cells going. But the multiple eccentric stimuli on a muscle is really going to be activating satellite cells. So you're probably, by increasing testosterone and IGF-1, first making the fibers grow by a direct stimulus, and secondly, you're tweaking the activated satellite cells and helping them proliferate and then fuse into muscle fibers. So you're secondarily adding more nuclei to the fiber that will then be stimulated to grow cytoplasm by testosterone and IGF-1.
Okay.
Right? So it's kind of a double layered impact.
Yeah.
Yeah. And there's no question that testosterone has a huge impact on muscle growth.
Right.
Now a certain proportion of the population doesn't necessarily want a ton of testosterone.
Yeah.
But it definitely is in there, and has a major important impact on muscle growth. But it isn't the full story, because there are times when the testosterone is low and you can still get growth. And certainly in women, you can certainly get muscle fiber growth and muscle mass increases with a reasonably low testosterone level.
Right. And that would almost take you back to the mechanical aspect of the mobilization of satellite cells.
Right.
just as important.
Yeah. There's that, always that component that I think people, if that's the one thing they get out of this, is that satellite cells are teeny little things that can have a big impact on the potential for growth and the future growth and the repair of muscle.
Right. Okay. All right, that's great. Thanks, Dr. Anderson. I mean, I probably have more questions than you want to continue to hear, but I learned a ton, and I want to thank you so much for being on the podcast here, and this was awesome. I was so excited to do this, and I really appreciate you doing this with me.
Okay, well, it just goes to show you that those early influences, thinking about biomechanics, can have a big impact on where your brain can go later, right?
Exactly.
So it's always fascinating.
Yeah.
Yeah.
Absolutely.
Good to speak with you, Dana.
Yeah, it was nice to meet you, and I'll email you after all this is done too. Thank you.
Okay, thank you.